engineering as a process depends on engineering DNA. In the lab, you guys will be using genetic engineering technologies. So the first one, recombinant DNA, uses restriction enzymes and ligations to make things. Secondly, PCR is used to amplify genetic material and change it. And last, automated sequencing is used to read out the DNA. So together these steps are known as genetic engineering. So what makes synthetic biology different are these three steps here. Firstly, automated construction of DNA. Here, instead of using recombinant DNA and PCR technologies, you pay somebody else and they make DNA for you. Nowadays, rather than cutting and pasting a DNA sequence out of an organism and into another, you can just type your DNA sequence into a computer or copy it from a database or even select it from a growing competent catalogue, and then you just order it over the internet. Secondly, we have standards. So to define the things you are building with standards, engineers want to treat DNA more like a programming language. Instead of ones, zeros, A, T, Gs and Cs, they want to use DNA to write a simple functional component.
Each fragment of DNA that we use, like Dana just said, we order it off the internet, most of it. These are called G blocks, and we can order them up to about 750 base pairs this year. And each of these fragments also needs a DNA overlap of about 20 to 40 base pairs to create the single DNA fragment. The really great thing about the Gibson assembly, and the reason we use it in our iGEM project, is it does only take an hour to form. Whereas recombinant DNA and genetic engineering, as Dana was just saying, can take up to months to do in a lab. It can take quite a while. So it's really fast. Okay, just a little bit about our project this year that we're doing. Okay, so we're trying to express chlorophyll in E. coli to eventually create photosystem 2, which would be the first step towards synthetically creating photosystem 2 to harness light energy in E. coli, and therefore green energy.